Hi guys, happy Thursday. It's awesome to be back here in the Street Science Lab to share another virtual classroom with you guys. And today we're actually learning about something that I did my master's in at university. For those of you who don't know me, my name is Jess, and at uni, when I was studying my Bachelor of Science with honours, I actually researched two different areas. One of them was botany, the other genetics. Now, does anyone know what those words mean? Think about it. No, I know what you're thinking. Botany is not the study of bottoms. That's something different, right? Botany is the study of plants. Think Botanic Gardens, Botany Bay, right? Botany is the study of plants. I think plants are absolutely amazing. Genetics, on the other hand, is something different. As a geneticist, what I study is something called DNA and hereditary traits. Now, today we will be exploring DNA, but before we dive too deeply in, we wanted to go through a couple of quick housekeeping things. Hello to everyone who's been hanging out with us before. We've got 160 of you in already today. Good morning. The rest are slowly getting out of bed, right? But for those of you who are fresh, let's quickly go over how our virtual classroom works. For starters, we have polls, and there is running currently a poll. You'll find a little tab somewhere on your screen, depending on what device you're using, and the polls tab open right now. We have a poll running where you can interact with us today. So right now we're asking, where are you tuning in from? Always stoked to see a few people from interstate, right? Overseas sometimes gets pretty exciting. But it seems today, so far, most people are tuning in from here in southeast Queensland. Hope it's a bit warmer where you are. It was freezing in my house this morning. As well as our polls, I would love to hear your questions about today. We are not doing it through the chat tab. Chats are disabled in this classroom. Instead, ask your responsible adult to tune into Street Science's Facebook page. You'll find a little post about what we're doing today, and I would love to see your questions there. My producer, Brian, will throw them at me during the show. Speaking of Facebook, those of you who've been here before may have noticed we had a different picture holding place before we opened up our classroom today. That photograph was one that was sent in to us on Facebook by Alina Chambers, and they were of our rock candy experiment we started a couple of weeks ago. Now, I'm not going to show you mine because mine didn't work, right? And since it didn't work when I wasn't here earlier in the week, somebody at Street Science actually cleaned up after me and threw away my experiment. Thank you to that person who cleaned up after me. And it's my own fault for not labeling my experiment. But for those of you who were looking, Alina's rock candy looked absolutely amazing. We had that perfect crystallization of sugar forming on those bamboo skewers. So well done, Alina and family. I hope you enjoy them today. Make sure you brush your teeth tonight and have plenty of water to counteract that sugar. I hope you enjoy it. Right, if you guys do have any experiments that you've done with Street Science, we always love seeing them on our Facebook page. And every day in the morning at Street Science, us presenters get together and share photos from our fans of experiments you've done. So please send them in. We'd love to see them. As well as Facebook questions during the session here today, I'll be jumping on later on to answer any more questions you have when I'm up in the office with a cup of tea. So we've talked about the polls. We've talked about our chatting and Facebook. I will mention quickly two special offers, right? In our special offers tab, you will find a link straight through to our Street Science web store. If you haven't already got your hands on one, you will be stoked to find that our Street Science Ultimate Junior Scientist sets are on sale again this week. I might have got the words jumbled up, but you know what I mean. The Ultimate kits have so much cool gear in them, and we'll be using some of that equipment today in our experiment. So jump online, order one of those if you haven't already. Last thing to get into, birthdays, right? We love celebrating our junior scientists here at Street Science, and I know it is Savannah's 9th birthday today. Happy birthday, you little legend, right? We are in interesting times. A party might not be happening, but let's party for you in our virtual classroom today, Savannah. Happy birthday, right? And thank you for my present for your birthday because we get to study genetics, something I'm very interested in. So now we know all that housekeeping out the way. let's revise quickly what you need for today's experiment because we're going to get started straight away. It does take quite a while to do. What we need is some fruit. Now, bananas and strawberries both work really well for this experiment. We'll talk about why later on. Right? I have a banana at home and I actually brought it in for my morning tea today, so I'm going to donate half of my banana to science. Also need a Ziploc bag or 2 would be better to help avoid messes. We need some salt, some water. Now, warm water or hot water works a bit better for this. Doesn't need to be super hot, not dangerous hot, but warm or hot water. Some dishwashing detergent or shampoo. What we need is something that has a chemical in it called sodium lauryl sulfate. Okay, sodium lauryl sulfate. But most of your dishwashing detergents and shampoos will have that because it helps break apart lipids, which is why we use it. You're going to need tablespoon and teaspoon measures and half a cup. Approximate. It's not a really important thing that our measures are correct today. And a stick for stirring, another cup for stirring as well. Now, guys, there is one more thing we need. And if you were paying attention to the directions, it said it needed to be cold. So mine's actually in the freezer. Excellent, Brian is grabbing it out for me. Right, doesn't need to be frozen, but just super cold. Thank you, Brian. Good man. Isopropyl alcohol. Now, I suggested you use isopropyl alcohol or ethanol. Both of these alcohols serve a really important purpose to help us purify our DNA in the last step. Isopropyl alcohol works a little better, so you don't need as much. Ethanol, you might need more. It doesn't need to be cool or cold, so make sure yours is chilling in the fridge or freezer for now. If you don't have isopropyl alcohol or ethanol, think again, double check you don't have any methylated spirits hanging out somewhere in the laundry cupboard. Because you'll find when you read the label that methylated spirits is actually 95% ethanol. It used to be methanol, but then they switched it to ethanol. It was a bit safer. Methylated spirits you probably have. If you don't even have that, I don't know what else you could use. Kelly's asked this question. Give a try with something else. If you've got the old vodka sitting around, you could give that a try as well. Best to keep that in the freezer anyway. But if you don't have it or it doesn't work today, by all means, next time you're at the shops, grab yourself some methylated spirits. It's handy to have at home anyway. Cool, let's dive right in. Genetics, the study of DNA. We're going to have a poll to help us out with this. DNA, or deoxyribonucleic acid, say it with me, deoxyribonucleic acid, it's a bit of a mouthful, that's why we call it DNA, is a super long molecule that's inside the cells of every living thing, right? That DNA has things on it called genes, with a G, not a J. And these genes define traits. Now, it's a bit complicated. So we've got an example in humans to help you get your head around it. Traits are things like hair color, like eye color, like how much melanin we have in our skin to get tans or not, and earlobe shape. And our little poll today is asking you which type of earlobe you have. Now, I've drawn a little picture here. I'm not very good at drawing. This person has an earlobe that hangs free, which means that this little hangy bit of the earlobe hangs below where it attaches to the face. You can sort of flip it around. This person has an earlobe that runs and attaches directly to the side of their cheek without any hangy flappy bits. Which type of earlobe do you have? We've got a poll running. I'd love to get your data here. If you don't know, run to a mirror quickly, have a look. Kind of hard to tell just by wiggling it around. Don't squeeze it too hard. They do have a few nerve endings out earlobes. I'm going to give it a couple more seconds, right, to do our first poll here. A biological study, a survey. Is your earlobe attached or unattached to your cheek? Forgive the typo there. All right, here we go. I'll give it a little bit longer. 3, 2, 1. Let's stop the poll. And I can see our results there. Of the 40, 50, 60, 75% of people who actually gave an answer that they knew, right? 41% have earlobes that are not directly attached to their cheek. 34% have earlobes that are are directly attached. And 23% of you didn't really know. Now, for those of you who don't know, I'd love for you to go have a look in the mirror later on and find out. But the odds are, the probability is that your earlobe probably hangs, is unattached. And that is because of DNA. The thing with DNA, right, is there are 2 copies of every gene. Those 2 copies, right, might be different to each other. They're called alleles. And sometimes one of those alleles is stronger than the other. We call that a dominant trait. Sometimes one of those alleles isn't as strong. It doesn't really express itself that much. And we call that recessive. Having earlobes that are directly attached to your face is a recessive trait. So if one of those alleles is the strong one, well, you never really see it, which means it's usually not as common. Now, obviously, there are a bunch of other factors that can affect the expression of traits, right? Meaning a lot of other things that can affect what we look like relative to our DNA, right? But earlobes is a really cool example. When you're going around the place, have a look at people's earlobes and see how many, do your own survey, how many people have attached earlobes versus hanging ones. Really cool, interesting study. And today, so far, slightly more people have unattached the dominant trait. Now, there are heaps of other traits we have that are defined by our DNA, but there are heaps of traits that are not defined by DNA, entirely anyway, like height, right? Adult height is something that we need the genes for to be really tall, but we also need to have the right environment growing up, the right type of food. I can say that my husband is really tall, but his parents, well, I would say a normal sized, a little bit taller than me. So how did he get so much taller than his parents that he can lean on their heads? Well, it's because his upbringing was different to theirs and he had different environmental factors helping his body grow from the blueprint or the design of that DNA. Now, humans aren't the ones that have DNA. I mentioned before, every living thing has DNA. Every living thing has traits. A little mouse might have bigger ears or smaller ears compared to its DNA. There are little birds, finches in the Galapagos Islands that evolved shorter and longer beaks, all because of DNA. And, well, our fruit has DNA as well. All plants have DNA in the nucleus of their cells. And our aim today is to try to pull that DNA out and have a look at it. Now, you can't just see it like this. It's absolutely tiny, right? Even if we got a regular microscope out, you couldn't see the DNA. It's so small. But there is so much of it in this banana. Every tiny little cell, every single cell has DNA inside it. So even though the DNA is so small, if we can mush it all together in one big lump, we should be able to see it. And to do that, we're using the objects you've got prepared. So let's get started, and we'll talk a bit more about it as we go because it takes a little while. I'm going to start with my banana. I'm going to peel it open. Now, I don't peel my bananas from this end. Check it out. Life hack. You break the other end. It's much easier and you're less likely to mash it. Monkeys do it this way. I'm going to break off about half of my banana and mush it into my bag. Now, guys, if you're using strawberries today, you want about 1/4 of a cup. Frozen ones actually work really well because the cells are already a bit broken. I'm squeezing a lot of the air out of my bag, and I'm going to seal it shut. And because I get a bit messy, my friends call me just the mess, I'm going to use 2 bags. Double bag it. I recommend you do as well. Squeezing the air out just means that your bags are less likely to pop open in the next step. So I recommend that as well. Squeezing out the air and sealing my second bag. Then we mash it. Now, I don't really need to show you guys this because you can guess what I'm doing, right? I have got my bag on the table and I'm pushing, mashing. I'll go on, I'll show you. It's kind of fun, right? You've had a long day, a long morning, a bad night's sleep. A little bit of mashing is always fun to make you feel better about it. Now, if you use a whole banana, right, you'll have a much better chance of getting lots of DNA, but I want to save some for my morning tea. Here we are now. I'm going to hold that up. You guys can see it's getting pretty mushed. We give it a little bit more And then, well, let's add something to it. Right now, I have mashed my banana, but I haven't broken the cells. Every living thing is made up of these tiny little parts called cells. Think of them as tiny, tiny miniature pieces of Lego all clicked together. Each cell has a bunch of different components inside it. And plant cells, unlike animal cells, have a wall around them, which means each cell is really strong. We need to break those cells. Now, freezing does a really good job of that because there's lots of water inside cells. And you guys know that when water freezes, it actually gets about 10% bigger, which is crazy, right? So those cells in that rigid wall, when the water freezes, it breaks that wall open. And when they defrost, well, the walls can't quite form again. So frozen strawberries, you're one step ahead. For our bananas, to help us break apart that wall, we're not going to use physics like forces or frozen water pushing on it. We're going to use a bit of chemistry. I'm going to get a fresh bead here, make a mess, and I'm going to measure out about half a cup of hot or warm water and about a teaspoon of salt. So we're making a salt water mix. Now this salt water is going to help us, right, with osmosis or lysing of cells. Osmosis is the diffusion of water across a semi-permeable membrane from an area of high concentration from an area of lower concentration. Lots of big words, but a lot of parents remember that one sentence from high school, right? But what I need you guys to realize today is that salty water will help fluids in the cells move or will help them bust out of membranes. The salt water will also do a really important job of helping to separate some of the natural materials, the chemicals, the proteins, the enzymes inside these banana cells, right? So our DNA will be more pure later on. And warm water just helps everything mix. I'm then going to pour, I'm not using a whole banana, right? I'm just going to pour a bit of my salty water in there and move it around. Use the whole half a cup if you need to. And really give it a good slosh. Sloshing it around. Now, guys, putting it in its second bag here for you would be really useful. I'm going to be gentle. And really make sure all that banana is mashed and the salty water is mixing through. Cool. Now that I've got a fairly homogeneous mixture, meaning one that is even, would you say that banana is dissolving in my salt water solution? No, I mean my salt's dissolved in my water, but you can still see those particles of banana suspended in my solution. So we have solid suspended in a liquid here. Is a suspension of a solution. We're going to move on to the next thing. Now, this will definitely help us break apart our cells, and it's going to do it chemically. The sodium lauryl sulfate, or our soap, serves a really important purpose of separating fats, right? Fats and proteins as well. And our cell walls are actually made up of a barricade of little molecules that have a fat at one end and a protein at the other. The fat part we call a lipid, right? This dish soap is going to do a really good job of busting apart all of those fats, breaking them open, so breaking a little hole in our wall. We're going to use about half a teaspoon of this, a good squirt. Pop that in there. I'm going to move all that air out so my bag doesn't pop open. And I'm going to gently swish that around again. Now, my aim here is to mix it. I don't want it to get really foamy, okay? So I'm being quite gentle as I make sure that that dish soap, right, mixes through with all of my banana solution. It's kind of fun, kind of therapeutic. Hope you're enjoying it at home if you are as well. Maybe you just spill tray if you're a bit worried of being a bit too violent with your bag and busting it open. Beautiful. And our next step, once we have lysed our cells with our detergent, is we're going to try to filter it out. Now, I've got one of our little tubes that you can get in our Ultimate Junior Scientist kit here. Falcon tubes are very useful things. We actually use these for DNA extraction in laboratories. right? I used to extract it from soil, from plant samples, using these exact tubes. Pretty cool stuff. I've also got a tray to keep my tube in. You can use a little shot glass or something. And I've made myself a little filtration system. Now, if you guys have paper towel, you can just stick it in the top of whatever cup you've got. Make sure it doesn't fall in completely. This is quite a strong hand towel I've got here. I want it to be quite thin. I'm going to wet it. Right, because I don't want it to absorb all of my banana solution. And I'm going to use a funnel. If you have one, get one. It does make it a bit easier. And I'm going to filter my banana solution. I'm going to stand up to do this so I don't make a mess in our lab, as usual. I'm going to pour my solution in, or my suspension, I should say. Now I can see that my liquid is making its way in. And I can hear you guys wondering, why are you putting it through paper towel? Isn't the paper towel going to trap all your DNA? No. Now that we've broken open our cells, right, the particles inside cells, our molecules we're talking about, are so tiny that they can very easily pass through our paper towel. The paper towel, though, is going to trap all of our chunks of banana and the cell walls as well. Beautiful. Now, this can take a little while. It might help to have a stick to give a little stir. Right, make sure you don't bust a hole in your paper towel, though. Awesome. Now, while I stir this, guys, have a quick look at the color of my solution. Is it still yellow? Sort of. It's also a little bit green, right? I see that the coloring from my dish detergent is actually passed through my filter as well. So you can see that this paper towel, while filtering out a lot of the, well, chunks and lumps that haven't been lysed, right, it also has enough color that is small enough to pass through my filter as well. Let's add a little bit more. Get as much in there as I can. Beautiful. I'll leave this there, and you can watch as I give it a little gentle stir to help it through. Now, this filter paper, as I mentioned, is filtering out, right, any unlysed cells. It's filtering out any large chunks of my banana, and passing through it is simply a solution containing our salty water still. Some of the salt would have been filtered out, but most of it's passed through, right? And our cell contents. I'm going to leave that off to the side. Might even try to filter out the rest of that into my salt water cup. I'm going to make sure I seal this bag shut because, well, we don't want anything going wrong with that leftover mashed banana. We don't want anyone accidentally trying to eat it. Of course they wouldn't. It's a laboratory. And can we see our DNA? No. Right, we've just got some like yucky coloured liquid. Right. What we have here, our cell contents, I've got a bit much. We need to get our DNA out of. It's still in there, in the solution, so small we can't see it. We want all of the DNA to clump together. And that's what our alcohol is for, right? The water that we have in our existing salt solution, right? Water has a really cool property of being polar, meaning one side of our water molecule has a positive charge, the other side has a negative charge. DNA also has that same property. The DNA molecules, they're really long, right? But they do have positive and negative charges, which means they dissolve really easily. Alcohol does not have a polar charge. So if DNA moves into alcohol, it will not dissolve and it's going to sit on top of precipitate in our alcohol solution. Now we have to be really careful at this step, right? We don't want to disrupt our layers too much, but we're going to try to get a layer of our isopropyl alcohol or ethanol, whatever you've got, sitting in the tube next to our salt water banana mixture solution. Here we go. I'm going to carefully squeeze it down pour it down the edge. If you have a pipette, right? That's going to be even better, or a syringe. It's going to be even better. Gently squeeze that on. Now you want about 2.5 to 5 centimeters of your isopropyl alcohol on top. You might need a little more if you've got ethanol. But when you pour it in, you're going to see, oh my goodness, it's already working for me. Now, I don't think you guys will be able to see mine, but I can see it already. I'm stoked. It's already working. You want to leave this here for like 3 to 10 minutes, depending on what you're using for your alcohol, how patient you are. But you're going to start to see the DNA precipitate, which means the DNA come out of solution and form into a solid clump. I can see these tiny little fingers, tiny little tendrils of white moving up into that layer of alcohol. Now, I doubt you'll be able to see it, but I'll lift it up to show you. You can see I have 2 distinct layers. My layer of banana mush at the bottom, right, and a clear layer of my alcohol on top. And I don't know if you guys can see. Brian, can you see a like white fuzz happening in there?
Yes.
Totally. Brian can see it from behind the camera. Hopefully you guys can as well. Just to the bottom of that solution, I've got a little bit of a white fuzz forming. That layer, that little bit of white fiber forming there is our DNA. Now, I said each molecule is super tiny, but they are quite long molecules. They're polymers, which means there are long chains of individual units all lined up. And when long chains get together in a liquid, they do tend to get a bit tangled, which means even though these tiny, tiny little molecules can't be seen with our eyes, when you get lots of them getting all tangled, they get all tangled up into one huge long line like a plate of spaghetti would, and you can see that huge clump, well, tiny clump, right, of even tinier individual particles. Now, this does take a while, right? You've got to wait up to 10 minutes for it to happen, but you can see it slowly happening. Tiny little tendrils at a time. While we wait for it to go, I'll set my timer. All right. Guys, if you're using strawberries, you may be having more success. And that's because plants, particularly ones that we've bred to eat, have these really interesting properties of having multiple copies of their DNA, right? Polyploid means to have multiple copies, right, of their chromosomes. Diploid means having 2 copies of each chromosome. Does that sound like any animals that you know? Yeah, having 2 copies. Pretty cool, huh? Right? But 3 copies, well, bananas have 3 copies of their chromosomes. That has happened because of the way us humans have bred them. Sure, it can happen naturally as well. Mutations occur in a lot of genes, which is how different animals and plants evolve, change over time. 3 copies of the chromosomes, like in our bananas, means we get a good amount of DNA. But strawberries, they have even more copies. No, not 4 copies, not 5, not 6, not 7. Strawberries have 8 copies of each chromosome. Isn't that wild? Right? So even though you might just have a tiny strawberry there, the number of DNA strands that can get tangled up from each cell is way higher. So you'll be able to see your DNA from strawberries much easier. So next time you get some reduced to clear strawberries, right? There are even some floating around at the greengrocers at the moment. Save a couple for another DNA extraction. You can try extracting DNA from any fruit or vegetable. Give it a go. Mash it up, something soft and squishy. If it's not soft and squishy, try pureeing it in a blender and put it through the same experiment. I would love to see the result you guys get from testing different fruits. Now, when you have got your DNA forming in the bottom of your tube, well, you can look at it in there, but you can try to get it out as well. It's only been about 5 minutes for me, 5-6 minutes, but I can see that it is forming in the bottom of my tube. We'll give it a little bit longer, right, and get ready to finish up our session. Guys, I do want to remind you that we love seeing you posting your photos and videos of your experiments on Facebook. So please share whatever you experiment with me today. I'd love to get your questions. And, well, if it's not exactly about DNA, I don't mind. I'd love your questions about anything about my beautiful plants or DNA. If you're interested in the store, don't forget you've got that Ultimate Junior Scientist kit you can get to play with at home with all these sorts of equipment. And, well, if you love getting hands-on doing science, we've got our STEAM lab as well, which we will pop up on our special offers page soon enough. Our STEAM lab are interactive workshops that our street science presenters do live with you and the kids, right? It is in your offers tab now, so check it out. In our STEAM lab, we've got one of our street science presenters, right, getting there with you, building something with you, giving you a list of experimental materials you need, and working through with your junior scientist questions as you go. So if you haven't signed up for STEAM Squad yet, definitely get on in the next couple of weeks. Sign up to a session. They are age level specific, right, and we tailor it to your kids exactly. I know we've had some really good feedback already. Now, the Learning Lab is another great resource if you guys are still learning from home and you need some more material, right? We have been developing lessons based on the curriculum for you there as well. So please jump on our Street Science Facebook page, on our website, check out our virtual learning options, because it's not just our virtual classrooms, guys. We'd love to keep teaching you elsewhere. I think I've bought about as much time as I can. It's time. Should we see if we can get my DNA out? Now, you guys can wait a bit longer, right? I'm going to try it now. I'm going to grab a bamboo skewer. You could use this paddle pop stick as well. And I'm going to carefully try to scoop up my DNA. I can see it, but will I be able to pick it up? Now, a twirling motion works well here, like spooling up a bit of thread. I can see it. It's working. It's working. The rougher the stick is, the better it will be for picking up. I can see a bit on that skewer, but I can see way more in the solution. I'm going to lift it up in a sec so you guys can see it, but for now, I'm going to focus. There we are. Paddle pop stick seems to work really well, swirling it around. I can't quite pick it up to show you guys. But if you can figure out a way of taking photos of this at home, please do. I know I've done it. You probably won't be able to see it, but I can see. Oh, no, you guys can't see it. There's a tiny little white droplet on the end of my paddle pop stick. I have got DNA out of a banana. Now, it doesn't translate really well on screen, guys, but I can see it in there, and I hope you guys can as well. Leave it for a little while, see how much you can collect, put it in a little dish, send us a photo, right? But congratulations to your scientists for extracting your very own DNA at home. If you've got any more questions about DNA, about bananas, about, I don't know, anything you like, I'll see you on Facebook later on today to answer them. I'm Jess from Street Science. I hope you guys have enjoyed this session today. Don't miss out on jumping to our web store or joining us on STEAM Squad soon. I'm Jess from Street Science. Have an absolutely awesome Thursday, and I think Steve-O will see you guys tomorrow. Same bat time, same bat channel for our next virtual classroom. Have an awesome day.
We love science so much here at Street Science. We teach kids about science, and we do it in a really, really cool way. We're going to be teaching the science of fireworks. We're going to be working with liquid nitrogen, some amazing visual stuff. It looks and feels just like smoke. Let's ramp things up a little bit. Who likes fireworks? I'm going to try and shoot these cups off your head. Not by using magic or anything crazy, just by using science that we can't see. Can you guys see a liquid in there? Look at that dark patch at the bottom. That's Laura's breath. Can we sneak in one more quick demo? We love science so much here at Street Science.